Today's lecture in Anatomy and Physiology is on fundamentals of nervous tissues. The outline of today's lecture is as follows. We will talk about white and gray matter. We will discuss parts of a neuron. Cell types in the nervous system will be discussed as well. We will also talk about the generation of an action potential, which is a nerve impulse. So we're talking about how nerves conduct signals. Nervous tissues can be classified as either white matter or gray matter. White matter are myelinated axons. Bundles of myelinated axons in the central nervous system are called tracts. Bundles of axons, myelinated or not, in the peripheral nervous system are called nerves. White matter is white because of myelin. Myelin is a fatty substance produced by glial cells to insulate axons and increase the speed of propagating action potentials, the nerve impulse, along an axon. Gray matter are the cell bodies. Collections of cell bodies within the central nervous system are called nuclei. Collections of cell bodies within the peripheral nervous system are called ganglia. Gray matter is gray due to the ribosomes in the cell body. These ribosomes are also called chromatophilic or Nissl substance. So let's look at the brain. You have your outer gray matter and your inner white matter. You have your myelin sheath, which is responsible for the formation of gray matter. Now let's discuss the locations of white and gray matter in the nervous system. In the brain, gray matter is located peripherally. White matter is located centrally. In the spinal cord, gray matter is located centrally. White matter is located peripherally. Let's take a look. The brain, your gray matter is peripheral, your white matter is central. Except for a few nuclei in the brain, this is generally true. The spinal cord, your white matter is peripheral, your gray matter is central. Let's look at the parts of a neuron. You have the dendrites, the cell body, and the axon. Your dendrites receive messages from other nerve cells. They're basically branch-like structures coming off the body. You have your cell body, which contains the nucleus and other vital structures for the life of the cell. And you have your axon, which is responsible for conducting a nerve impulse down the cell. Notice that this axon is myelinated. Now, the different types of cells of the nervous system. We already talked a little bit about neurons. They send nerve impulses. We also have glial cells, or neuroglia, which literally means nerve glue. These are the supporting cells, and they're more abundant than neurons. In the central nervous system, astrocytes and oligodendrocytes are the most abundant. Astrocytes, as its name suggests, are star-shaped cells. These are general supporting cells. So this is an astrocyte right here. You have your oligodendrocytes, which are cells with multiple processes which myelinate multiple neurons. So this would be your oligodendrocyte. And look at this oligodendrocyte here, myelinating multiple neurons. You have your microglia, which are from the blood monocytes, and they become macrophages in the presence of infection or injury. So essentially, unless there's a major injury to the brain or a major infection in the brain, you barely see these cells. Ependymal cells are the cells that line the ventricles of the brain and produce cerebrospinal fluid. In the peripheral nervous system, your glial cells consist of Schwann cells, which myelinate axons. So this is your Schwann cell. It's similar to oligodendrocytes, except Schwann cells only myelinate one neuron at a time. Satellite cells surround the cell bodies and provide support. That's why they're called satellite cells. They circle around the cell. Now let's talk about generation of an action potential, also known as a nerve impulse. So neurons have something called a resting membrane potential, which is basically the electric charge. So the inside of a cell membrane is negative relative to the outside. And this number is normally around minus 70 millivolts, but it can fluctuate between between minus 40 millivolts and minus 90 millivolts. And this is due to the presence of more negative ions in the cell. And this resting membrane potential is maintained by sodium channels, potassium channels, and sodium-potassium ATPase pumps. And in addition to resting membrane potential, there are two types of potentials. You have your graded potential, which are small changes in the membrane potential, which get weaker with time and distance along the cell membrane. So as more time passes, this graded potential gets weakened, and the further it gets away from the center of the cell, the weaker it gets. Action potentials are large specific changes in membrane potential, which maintain their intensity with time and distance along the cell membrane. Excitable cells are able to respond to stimuli to generate graded potentials. Conductive cells are able to propagate an action potential. So nerve cells are both excitable and conductive. So nerve cells can respond to another nerve impulse and generate a nerve impulse of its own. So let's look at the generation of an action potential. And feel free to refer to this graph. So the nerve cell generates a graded potential in response to a stimulus, which initiates depolarization. And when I say depolarization, it means the membrane potential becomes more positive. So basically you have a nerve cell, it gets stimulated by another nerve cell, and it generates a graded potential. This graded potential acts as a switch, and once a threshold is reached, usually around minus 55 millivolts, an action potential is generated. So you got your action potential, and right here, your fast-gated sodium channels are opened, which lead to rapid depolarization. So the membrane potential becomes more positive. And these channels quickly close, which are why they're called fast voltage-gated channels, because they close fast. And sodium enters the cell, which is why the membrane potential becomes more positive. After this, slow voltage-gated potassium channels open after the peak of the action potential is reached. And as a result, potassium diffuses out of the cell, which leads to repolarization because the cell is becoming more negative. The resting membrane potential is becoming more negative because positive ions are leaving the cell. So you got your repolarization followed by hyperpolarization, which is when the membrane potential becomes more negative than the resting membrane potential of minus 70 millivolts. After that, there is the return to a resting membrane potential. So in short, there is a stimulus which causes a nerve cell to generate a graded potential. This graded potential acts as a switch, a threshold is reached, and an action potential is generated by opening fast sodium channels and slow potassium channels, causing rapid depolarization, then repolarization, followed by hyperpolarization. The arrestin membrane potential then returns to baseline. This action potential nerve impulse can travel long distances, and this is made possible by the myelination of axons. It prevents the leak of ions and increases the conduction speed by up to 200 times. Just think of the axons as an insulated electric cord. The end. It looks like we're done. Like this video, subscribe. Thank you.